session is about the microbiome therapeutics. And I am Doyomni from Seoul National University, and the organizer and the chair for the first part. And second part will be gone by the Professor Jin Byung Park from Women's University. I would like to introduce the first speaker, Professor Ji-hyun Kim. And he is currently the professor of Yonsei University. And brief his academic record, he is graduated from the Seoul National University for undergraduate and master and a PhD. He has had very many awards and also the very many great publications. And today he is talking about the current status and future prospect of research and development on microbiome therapeutics. Please give a warm welcome to Professor Ji-hyun Kim.
Morning, everyone. It's an honor to be here, and before I give my talk, I would express my sincere thanks to Professor Jin Byung Park and also Professor Doyeok Lee for inviting me to give me an opportunity to present my view on the current status and future prospects of microbiome therapeutics. So let me start. All of you know who these people are, and this is the title I just briefly talked about. And in microbiology, the great discoveries have accompanied with the development of technological tools. For example, the microbial world was first discovered after the microscope was invented, and also through pure culture techniques, we now can study many microbes for their roles in human health and others. And in recent years, actually there is another technology, high sequencing technology that enables us to look into the genetic complement of microbes in the environment. So let me just briefly give you the definition again. The microbiome actually is constituted of the microbe itself, which is called microbiota, and the genetic complement, metagenome, and more. I'll talk about that later a little bit more. But anyway, since the turn of the century, actually we are now having very powerful tool, so-called next generation sequencing technologies, or the third or the fourth, that can give us a vast amount of genetic information to deal with. So the term microbiome has been redefined in recent years to include that not just the microbes themselves, but also the diverse products it can make, from nucleic acids to proteins to lipids, saccharides, and even small molecules. And finally, the environment itself can be considered as part of the microbiome. So this is called theater of activity. So what I'm talking here is that microbiome, the concept is very big. So when you want to say something specific, for example, the microbes, you need to use better term. And all these are part of the biome that constitute the biosphere on Earth. Actually, microbiome field and also the microbiology itself is changing very rapidly. With many tools available now we can have an access to those microbes and also microbial information. So before that, actually, we almost always depended on cultivation. We just saw very small fraction of the whole microbiome. But now, with the help of brute force technologies, such as metagenomic tools, now we can have an access to what kind of microbes are there and also what kind of genes they have to function in various roles. And also, we can monitor the gene expression, protein production, and also molecular catalysis. So with these big changes, actually, what drove this trend was big consortium activities from the United States and also the European Union and other countries, including China. They have had a big census type microbiome analysis. And that helped us a lot. And also individual researchers walk into individual targets of health and disease to know that there are many diseases that we never thought about before are actually influenced by microbes. First, there were obesity and diabetes, and then from those metabolic diseases to immune diseases and even cancers and neuro or heart-related diseases. Nowadays, we realize that there are almost every disease that are from which microbes are involved in. So these microbes in our system, especially in the gut, are affected by many factors. Of course, we can think about our genetic factor, but microbiome is influenced more from the diet you have and also the lifestyle or hygiene you have. From these effects, actually, the microbiome can change from normal state to abnormal, and it causes chronic inflammation, which can be the cause of many other diseases. So here we have another concept called dysbiosis, and different from homeostasis or eubiosis, which can be considered as in a coherent form, but this dysbiosis can be anything from that eubiosis. So this term, again, just like microbiome, you need to be very careful when you use this term. So you need to have some specific description when you talk about dysbiosis. Actually, from many studies in recent years, now we have more candidates for use in human health. Previously, as you know, lactic acid bacteria and bifidobacteria have been used for probiotics. But nowadays, we have more candidates such as Akkermansia or Faecalibacterium. Well, I'm not saying these bacteria are good all the time, but at least in some cases, these bacteria can be utilized for improving health. And going from there, actually, people are now seeking a way to treat those diseases affected by microbes. The first indication came from fecal microbiota transplantation to cure Clostridioides difficile infection, CDI in short. In fact, it was so effective here. no laser pointer? It's okay. So on the left top, it shows the effect of FMT is even better than normal antibiotics. So because of that, there were many clinical trials to use gut microbes for treating CDI. And last year and this year, we now have actual drugs approved by FDA. But that's just tip of the ice, actually. Nowadays, many more diseases, including several cancers and also immune-related diseases. Now there are trials for their use in human health and treating disease. So basically, these microbiome therapeutics can be divided into several categories. First of all, as I just told you, FMT itself can be used, but there is a safety issue there. So another possibility is to collect some key microbes or keystone microbes and then use that as collection. And also, there are other possibilities, including using the microbial products and also even utilizing the viruses that infect those problematic microbes. So let me give you just a few examples how we are looking into these microbes. First example is gastric cancer and Helicobacter pylori. And as you all know, the famous Korea's hypothesis dictates that from normal microbiota or normal mucosa, once it is infected by Helicobacter pylori, it causes gastritis. And this chronic gastritis can be developed into intestinal metaplasia and dysplasia, eventually gastric cancer. And before Helicobacter pylori was known, actually people thought that nothing can survive in the stomach. But with the accomplishment of Barry Marshall, who won Nobel Prize, actually this Helicobacter bacterium can cause gastritis. But that was about it. But in recent years, now we realize that there are many more bacteria in our stomach, and they can affect our health. So we embarked on an experiment using germ-free mice in which we collected biopsy samples from gastric cancer patients and then transplanted to the germ-free mouse. And after about a year, actually we observed that these humanized mice can develop into gastric cancer. This is surprising. Why? First of all, Helicobacter pylori cannot survive in mouse stomach. So this indicates that some bacteria from human stomach that is not Helicobacter pylori can have the ability to cause cancer. So this is against the current dogma. And also, we believe that Helicobacter itself may not be the cause of gastric cancer, although it can cause gastritis. So now we are actively looking into what kind of microbes are responsible for gastric cancer, and we have some human cohort data. By analyzing them in various ways, we were able to define some bacteria that are associated with healthy state and other bacteria that are associated with gastric cancer. And the examples shown here are the ones that might be responsible for gastric eubiosis and also maintaining health. And the indication using germ-free mice is that actually these bacteria can fight against Helicobacter pylori, another bacterial species that can survive or thrive in the mouse stomach. And that also causes gastritis. And what we saw was the reduction of gastritis and other disease using this microbial cocktail. And also, we found some microbes that are enriched in gastric cancer. And when we administer these bacteria individually or collectively into germ-free mice, actually they caused hyperplasia. So some of these microbes, in fact, might be responsible for the gastric cancer, not Helicobacter pylori. Another example is on colorectal cancer. Here we have two approaches. One is, well, when patients are first diagnosed with colorectal cancer, they usually, through biopsy, actually, polyps can be removed easily, and or surgically removed the cancer area. But the issue here is, well, they can go through chemotherapy too. The issue here is, for some patients, after those treatments, they recover their health. But others are not so fortunate that they eventually have to pass away. So we wanted to know whether microbes can be a predictor for those outcomes. So we analyzed the microbiota from patients who are first diagnosed with colorectal cancer. And then by analyzing them, we were able to define which bacteria are good or which bacteria are bad. And we developed a marker out of it so that after 3 or 4 or 5 years, whether that person can survive out of this cancer or not. And actually this marker was very efficient and outperformed existing other biomarkers or markers that related to microbiota. In any case, this can be used right now in medicine, I think. So if there is no bacteria in the gut that can be used for these purposes, for good or bad, actually we can make it. So through these synthetic biology approaches, we can produce designer probiotics. And this is the case that I collaborated with a Korean company called 쑨원대학교. And here we made lactic acid bacterium that can produce anti-cancer peptide. And when we applied that to mice, actually the cancer size was reduced in 2 mice models. And also, one thing interesting was the microbiota status was changed from dysbiotic to eubiotic. So this antibiotic producing probiotic can not only work against colorectal cancer, but also can improve the gut microbiota status. Well, this kind of therapeutics can go farther than traditionally known diseases, such as aging that I'm interested in right now. So the first observation we had was actually there were subtle differences between young people and old people that other people did not notice. So we had a hypothesis that, well, if that's the case, then if we move the microbiota from the young to the old, there could be a chance that the old can become younger even, or at least the aging process can be slowed. And that's in fact what we observed. And when we used 4-week-old mice and 12-month-old mice and 25-month-old mice, which correspond to about 15 or 20 years of age in human, or 45 to 55, or more than 80 in humans. So we had young, old, and very old models, and then we transplanted the microbiota from this young to old, and what we observed was, in fact, the old mice becomes stronger because they have higher muscle content, and also their skins are tighter and is able to hold more water in their skin. And we looked into the mechanism and briefly and through RNA-seq, and when we checked the gene expression in the skin and muscle, we noticed one of the, well, there are other genes that are very interesting right now, but we picked one gene, and we were lucky that to have mice that are heterologous in this gene called DBN1. And when we compare those mouse strain with the strain that has these 2 copies of this gene, actually the homotype can have better skin properties. And also we now have an idea what kind of microbes are responsible for that. So giving you several examples, what I told you was we are now entering into the micro universe that we never thought of, and this impact will be huge, and there will be many challenges ahead of us. The first thing is, well, people think that, well, we know much about healthy human now. That's not the case. Actually, even in the Western world, that's the case. For Koreans, that kind of study has not been done. So we need to do that. If you want to have good pharmaceutical, microbiome-based pharmaceutical, you need to know what is normal and abnormal. So we take it granted, but it's not. So we need to have more healthy human data. Tens of thousands or hundreds of thousands of people need to be more analyzed. And also, because the history of this microbiome study is very short, we do not have yet longitudinal data, at least 5 to 10 years of monitoring for one subject. Of course, we need to have better view. The next speakers will talk in detail about this. If you want to know the mechanism, you need to have a holistic view, not just one microbe, microbiome, and also human factors, environmental factors, and also to develop a good drug, knowing the mode of action is critical in these years. So that's something we have to keep in mind too. And also currently the targets are mostly bacteria, but in the gut there are other microbial life forms such as fungi or, well, I'm not sure whether we can say life for this one, but anyway, viruses, and also even larger animals survive in the gut. So we need to take these things account. And finally, we need to have a better tool to isolate these bacteria. As I told you, less than 1% of the bacteria or microbes in the environment can be cultured in the lab currently. So we need to have better information about their physiology and their preferences. Well, this is something I already talked about. We have many tools right now to attack pathogens, pinpointed. But anyway, we will have more ways to come to be used as microbiome therapeutics. Thank you very much for hearing.
Thank you very much, Professor Kim. And due to the limited times, we all would like to have one or two questions from floor. Yeah. Do we have a mic? Oh, okay. You can stand over there and then ask a question.
So that's a very nice presentation. So I noticed in your one of your projects you used gastric microbiota transplantation, and the other used young mice microbiota transplanted to the old mice. Did you find any specific bacteria that were functional in terms of ameliorating?
Yeah, we do. Because of patenting issues, I didn't specify those bacteria, but we do have a collection of microbes from healthy stomach that can be used as pharmaceutical candidates. We tested those microbes using germ-free and SPF mice, and it can prevent gastric illness.
Yeah, cool.
And also for the aging part, we do right now have bacteria, but one of them was not cultured yet, but still we know what kind of bacteria are responsible for that.
So the other question that I want to ask is, when you do gastric microbiota transplantation, did you do any measures to enhance your acidic pH in the gastric so that the bacteria can be alive in there?
Yeah, actually that's a very good question. Well, even in the case of Helicobacter pylori, it cannot survive at pH 2. The reason why Helicobacter thrive in the stomach is because they can go into the mucin layer, and then from there they produce molecules that can counteract acidic molecule. So that's the secret of that bacterium. And many other bacteria, including Akkermansia, have that kind of ability. And some others are, to some degree, well, as you know, acid tolerant. So in that gastric microbiota transplantation, we didn't do anything. We just,
Yeah. So is the bacteria still alive after you do?
Yeah. We monitor that. Yeah.
Yeah, cool.
And it can be found in the feces, and it can be found in the stomach after we sacrifice those mice, and we check that.
Cool. Thank you. Fantastic study.
Thank you.
Due to the time limit, we will have a further question after this session if you have. Thank you very much, Professor Kim. I would like to introduce the second speaker, Dr. Myung-hee Kim. She is currently the director and the principal and research scientist in the CRIP, the best national lab and then institute in Korea. She also has had very many good and reputational publications, including the recent prominent paper on the amino acid tRNA synthesis. That is I guess the today's main topic. And please give her the warm welcome too.
이제 된 것 같아요. 네. 네, 된 것 같아요. 네. First of all, thank you so much for the organizer for this session, Professor Do-Yeon Lee and Byung-Jin Park for inviting me this very nice symposium within the Big C 2023. So 시작해도 돼요? 네. So today my talk will be focused on a gut-associated bacterial tRNA synthesis, which may be a therapeutic target for inflammatory disease. Many studies showed that gut microbiota influences the function of organs. Thus, it is now being established that disease states are correlated with imbalances in the gut microbiota. Particularly, a number of studies highlight that the gut microbiota shapes immune and determines our health conditions. Thus, the gut microbiota is considered the most significant part in maintaining gut health. So my lab has studied on human tRNA synthesases for a long time, maybe more than 15 years. So as you know, amino acid tRNA synthase, in short ARS, is a ubiquitously expressed enzyme that charges a specific amino acid to the cognate tRNA for protein synthesis in every living species. During the evolution of higher eukaryotic systems, the ARSs acquire an additional domains and regions. So this emergence of such structural additions is linked to non-canonical function beyond protein synthesis. So the functions of human ARSs cover a broad range of cell signaling, including transcription and translation regulations. These suggest that human ARSs are mediators to maintain the homeostasis. For these reasons, some of human ARSs have been developed for therapeutics for many diseases, including cancers. So in this regard, we hypothesize that in addition to human tRNA synthetases, commensal tRNA synthetases in the gut microbiota also can act as mediators to maintain the homeostasis. We particularly paid attention to the gut-associated bacterium, Akkermansia mucinipilla, just Dr. Jihyun Kim mentioned. This bacterium is interestingly threonine autotroph. Thus, this bacterium has to acquire the amino acid to survive from threonine-rich mucous layer on the intestinal tract. That is why this bacterium's name is mucinipilla. Importantly, This bacterium is negatively correlated with many immune-related disorders, including inflammatory bowel disease, IBD. So we questioned on involvement Akkermansia mycinipilla ARSs in the immune regulation. So Sinhe Park, PhD student in my lab, purified all ARSs from Akkermansia mycinipilla and evaluate the immune regulation of the purified ARSs in mouse macrophages, BMDMs. Interestingly, only tRNA tRNA synthetases triggered the representative anti-inflammatory cytokine interleukin 10. Then she also evaluated the other conventional bacterial tRNA TARS, tRNA tRNA synthetases, and as well as human TARS, and observed no immune regulation function of these proteins. Thus, these results suggest that the AM TARS function is very specific in immune regulation. Akkermansia mycinipilla extracellularly secretes AM TARS rather than via extracellular vesicles to play a role in the immune regulation under normal steady state condition. AM TARS triggered much more significant level of IL-10 in the LPS-treated inflammation conditions in both mouse and human macrophages, and even significantly reduced suppressed pro-inflammatory cytokines IL-6 and TNF-alpha. Recently, multi-omics database of microbiome from IBD patient and non-IBD controls were reported and publicly opened. Because this IBD is the most presentative inflammatory disease, we can test. So we utilize this database to evaluate the correlation between the abundance of Akkermansia mycinipilla and IBD. Constant with previous reports, metagenomic analysis revealed that the abundance of Akkermansia mycinipilla is significantly lower in IBD patients. Further functional profiling revealed that the abundance of total bacterial TARS is not significantly lower in subjects with IBD. However, the level of AM TARS is significantly lower in IBD patients. The levels of other ARSs from Akkermansia mycinipilla also lower in subjects with IBD, suggesting that the levels of Akkermansia mycinipilla tRNA synthetases associated with the abundance of Akkermansia mycinipilla. Similar result was seen in meta-transcriptomic analysis. Thus, these data suggest that a reduced abundance of Akkermansia mycinipilla in IBD patients is associated with lower levels of AM TARS. This may lead to dysregulation of AM TARS mediated anti-inflammatory immune responses in the subject with IBD. To evaluate this, our hypothesis, we evaluate the anti-inflammatory function of AM TARS using the SS induced colitis mouse model. Intraperitoneal IP injection of AM TARS significantly improved disruption of the epithelium lining and damage score in the colon, thus reduced disease activity in the colitis mice. Then what is the target immune cell of AM TARS? To identify the cells targeted by the AM TARS, mice were IP injected with fluorescently labeled AM TARS, and it was detected predominantly on macrophages rather than T and B cell. Furthermore, we observed that the administered AM TARS co-localizes with macrophages, mainly in the intestinal lamina propria, and further detect significantly increased IL-10 in the PEG and intestine-derived macrophages. Thus, these results suggest that AM TARS target macrophages in the gut. We further analyzed that AM TARS promotes M2 macrophages activation, linking to IL-10 production under both normal condition and inflammation conditions. Thus, we observed that AM TARS treatment significantly lowered colitis-induced inflammatory monocyte cells and restored IL-10 positive macrophages, and consequently increased the IL-10 levels in the mouse serum. Then what is the mechanism underlying the AM TARS immune regulation function? To elucidate the molecular mechanism underlying that function, we compared its amino acid sequence with those of other TARSs. As you can see, AM TARS contains unique 1,2 region, which within its N-terminal and catalytic domains, which are not found in other TARS. We modeled the structure of AM TARS using the famous Alpha-2 software. The structure model suggests that both U1, U2 unique regions within AM TARS are surface exposed and may be involved in the interaction with host target, such as signaling receptors. We evaluate the functionality of unique regions U1, U2. Deletion of these unique regions did not affect its enzymatic activity, but abolished the IL-10 production in BMDMs. Consequently, these mutants did not recover the pathological activity in the colitis mice. Thus, these data clearly demonstrate that the unique regions within AM TARS are essential for the anti-inflammatory immune function. Then what is the host target of this AM TARS? To identify the host target of AM TARS, we performed membrane protein interactome analysis using human macrophage THP1 cells. To make a long story short, AM TARS only interact with toll-like receptor 2, not with toll-like receptor 4 and other membrane receptors. We evaluate the specific direct interaction between AM TARS and toll-like receptor 2. As you can see, at the protein levels, AM TARS directly interact with toll-like 2. And we further evaluate the toll-like receptor dependency for AM-TARS function. As you can see, we used BMDM from isolate from toll-like receptor 2 knockout mouse. Without these unit regions, AM-TARS did not function in the immune regulations. We further confirmed the requirement of the unique regions for the toll-like receptor 2 dependent immune function of AM-TARS by engineering E. coli TARS. You remember this data in the beginning of my presentation? E. coli TARS had no anti-inflammatory immune function in BMDMs. In cooperation of the AM-TARS U1, U2 sequences into the corresponding regions in E. coli TARS compared the ability to trigger IL-10 production on E. coli TARS via the interaction with toll-like receptor 2. However, IL-10 production by E. coli, yes. So these function further assessed in the role of this function in vivo using toll-like receptor 2 knockout mice. Colitis-induced disruption of lining, the epithelial lining, and the damage scores in toll-like receptor 2 knockout mice did not improve after AM-TARS treatment. Then what is the signaling pathway after toll-like receptor 2 interacting with this TARS, AM-TARS? To identify the signaling pathway triggered by AM-TARS, we assayed kinase inhibitors and conducted immunoblotting assays. To make a long story short, the interaction between toll-like receptor 2 with AM-TARS actually activates transcriptional factor CREB, which is most critical for IL-10 production via MEP kinase and both MEP kinase and PI3 kinase AKT pathways. We furthermore clarified the signaling using the NF-kappa B transgenic zebrafish model. It clearly showed that the central inflammatory mediator NF-kappa B was significantly increased in the colitis zebrafish model. But the treatment of AM-TARS significantly lowered in the colitis zebrafish for the anti-inflammatory function of this result for the validate the anti-inflammatory function of AM-TARS. Then finally, we engineered the famous probiotic E. coli Nissl to deliver AM-TARS in vivo. So we developed the engineered probiotics, chromosomally input the AM-TARS gene. The engineered E. coli Nissl was secrete AM-TARS extracellularly at significant level, but much more level of AM-TARS from Akkermansia myciniphylla. Nevertheless, it colonized and secreted AM-TARS, and the secreted AM-TARS localized with macrophages in the intestine. Its oral administration significantly restored IL-10 secreting macrophages and markedly increased IL-10 levels in the serum of the colitis mice. and improved chloride-induced disruption of the epithelial lining, damage scores, and disease activity score. So, in summary, in this study, our result provides evidence that the gut-associated bacterium, Akkermansia muciniphylla, secretes AM TARS to monitor and modulate host immune homeostasis. Thus, our study highlights that commensal tRNA synthetases may act as instinct mediators shaping holobiont homeostasis. Since AM TARS modulates intestinal inflammation, it might be able to control systemic inflammation in disease. Thus, conventional tRNA synthetases may be new strategies for the prevention and therapeutic treatment of various inflammation disease, including IBD. So many people were involved in this project to succeed this story. I thank everyone in my lab and also many scientists in my institute, and also Wonjeri, Seoul National University, and also Gwangpyo Go to analyze the bioinformatics data at Seoul National University. Thank you.
Thank you very much. And we are going to have some question from floors first. Yeah.
Thank you very much for your great talk. So it's very interesting and very informative. I have a question about AmTARS. AmTARS is produced in cytoplasm, right? In Akkermansia.
Yes, within Akkermansia muciniphylla.
Yeah.
Yes.
Then AmTARS is going to be exposed to macrophages after cell lysis, right?
No.
No?
It directly secreted extracellular.
Secreted enzyme?
Yes. But actually, in case of human tRNA synthetases, some human tRNA synthetases are secret to do function. But we don't know what is the mechanism of this secretion yet.
Okay.
So this bacterium also secrete directly extracellularly and do a function.
One more question is that
Yes.
Akkermansia was known to produce a number of biologically active metabolites,
Right.
such as short-chain fatty acids.
That's right. Yes.
And peptides and proteins, probably the polysaccharides. So how would you expect they interaction
This.
in host cells?
Very nice questions. But actually, you remember this Akkermansia muciniphylla is threonine autotroph. So it must acquire the amino acid from mucin. So actually, we try to clue the correlation of this threonine autotroph and secretion of this AM TARS. But yet, we actually did not have a promising clue between the two molecules, threonine and AM TARS. But anyway, that's the one of question we must address in the future. And other molecules like short-chain fatty acid and other amino acid metabolite, actually we don't know.
Okay, thank you.
This is really a great study. I've asked my lab to read your paper. So we studied in length. But you know, one question that I'm very interested in, how did you find the receptor is TLR2, toll-like receptor 2? You know, the TLR2, the ligand for TLR2 is usually lipoprotein, but for synthesis, tRNA synthesis, how did you link that to TLR2 rather than other receptors?
But actually, we used human macrophage cell, THP1, and we pull down the purified TARS using the THP1 cell lysage, but membrane parts, and then do proteomic analysis. Then we actually had quite few candidates, which are all receptors to trigger these immune responses, but only toll-like receptor 2 interact with TARS, AM-TARS, not toll-like receptor 4. Actually, even toll-like receptor 4 not in the list as a candidate. That's the how we identify the human target toll-like receptor 2 using AM-TARS.
Cool. Thank you.
I have a quick question.
Okay.
Is there any further evidence to the interaction, physical and structural interaction between the toll-like 2 and the mTARS?
That's also a very nice question. Actually, the binding affinity between AM-TARS and toll-like receptor 2 is not strong.
Oh, okay.
Yeah. I guess this is true because if the affinity between two proteins are strong,
Right.
then the cell system would be disrupted.
Okay.
So that's why, even though our lab is based on structural biology, we didn't try to solve this complex structure directly.
Okay.
Yes.
Okay. Thank you very much for your talk, Dr. Kim.
Thank you.
Okay, we are going for the third speakers, and I would like to introduce the Professor Tao Zhou, and he is currently the professor at Sun Yat-sen University, and then also the associate director at the Sun Yat University affiliated hospital. Despite his young age, it's right, you're relatively young, and despite the young ages, he is already the leading this field, particularly on the gut virome and also the fungum, as we can see from the title of his talk, and he's going to talk about the role of gut virome in gastrointestinal disease and fecal microbiota transplantation today. Please give him a warm welcome, and then he will start the talk.
Thank you, Professor Lee. So today I'll share you some work and the story on gut virome in gastrointestinal disease and fecal microbiota transplantation. My name is Tao Zhou. I'm from Sun Yat-sen University in China. And our lab's interest is mostly in the non-bacterial microbiome part and their roles in GI diseases. And as everyone knows, the bacteria plays a very important role in human health. But in our gut, we also harbor a great number of viruses and phages and beyond, such as fungi. And the viruses and phages, the total amount is almost 10 times higher than that of bacteria. So today, my story will be mainly focused on the gut virome. Now, the first and foremost question is, what is the gut virome and what is the composition? And mostly in our gut, we have the viruses and phages. And the phages, most of them are cadaverolius and macroviridae. For this phages, it constitutes more than 95% of our human gut virome, while only less than 5% of our gut viruses are eukaryotic viruses. And then at the time, about 4 years ago, we wanted to study what is the composition of gut virus in healthy humans. We started with healthy Chinese and recruited more than 900 healthy Chinese and studied their gut virome. And we recruited subjects in Yunnan province. This is a very interesting district because we have in Kunming, which is an urban city, the right ones, which is an urban city, we have multi-ethnicity cohabitance. Well, in each areas, we have different ethnic minorities residing in respective regions. So we studied the virome. On the right side, you can see that the virome is actually forming a continuum, which means the gut virome varies substantially across healthy individuals. And then we separate the healthy Chinese into rural residents and urban residents, different ethnic minorities. And for ethnic Han and ethnic Zhang, the gut virome is actually the diversity very low. Then after looking at the urban counterparts, you could see their microbiome, the virome is diversified again, which means multi-ethnicity cohabiting could homogenize the gut virome and increase the gut virome's diversity. And then we ranked the metadata, the factors that will actually has a big impact on the gut virome, and sequentially they are geography, where the lives, and dietary habits and medication, and then urbanization. And then following the composition of the gut virome, you want to ask how does the gut virome work and its function. So absolutely, the gut virome functions through different layers. The first layer is that the virus, especially eukaryotic virus, can directly interact with the mammalian host, which is in the context of gut. It can interact with the gut epithelium and immune system. And for the primary component of the gut virome, which are bacteriophages, they can interact with, bacteria, and through interacting with the bacterial microbiome, it functions. Then we have a lot of viral sensors in our gut, just like toll-like receptors and RIG-I and C-gas. They are DNA and RNA sensors. The viruses and phages can release DNA and RNA, which can be sensed by the mammalian host. And then it comes to fecal microbiota transplantation, because most of the fecal microtransplantation studies were based on gut bacteria. And our understanding is that fecal microbiota transplantation can cure a disease by just replacing the bacteria. And it has been used in many diseases, but the primary disease that is employed in is C. difficile infection. And C. difficile infection, just like Professor King talked about it. CDI disease is a diarrhea disease, mainly involved in GI inflammation. And antibiotic therapy is always the first line therapy, but up to 1/3 of the patients do not respond to this therapy. So we choose to use FMT. Then FMT is highly effective in treating recurrent CDI, and most of our understanding is on the bacteria, on the restoration of the bacterial microbiome. Then how about the virome or the phage? Again, about 5 years ago, Professor Stefan Streiber published a paper. Even though this is an exploratory study using only 5 cases, they use the sterile fecal filter transfer, which means they remove the bacteria and only use the sterile filtrate. But the sterile filtrate is still effective in treating these 5 patients, which lead us to hypothesize either it's because the metabolism or because the viruses are phages, because viruses and phages cannot be filtrated just using a 0.2 millimeter micro filter. Then we want to study whether fecal virome is affecting the FMT efficacy. And then it comes down to the gut virome in fecal microbiota transplantation. And two questions that we want to answer through this study is, the first, whether there is a virome dysbiosis in CDI, then whether FMT can restore the gut virome. And then let's look at the virome dysbiosis in CDI. And we interrogated the fecal virome at the time in CDI, and we found a significant increase of cultivarulose bacteriophages, which is double-stranded DNA phage order. But though the total abundance is increased, but the virome richness, diversity, evenness were actually decreasing in CDI. Then, as to other viral families, we found macroviridi, which is a single-stranded DNA phage family, and then anaeroviridi, which is a family of eukaryotic viruses. This family of viruses will actually increase because in CDI they definitely have an immune suppressed state. So therefore, the eukaryotic viruses may harness this chance to bloom. And then we looked at whether FMT can restore the gut virome. And at the time, we only have 9 patients. 6 of them were cured. Then 3 of them had a relapse, especially for the last patient, patient 9, it has a late relapse. And overall, the FMT restored the gut virome, especially at the baseline, you have a high abundance of cultivarulose, then it was decreased by FMT. And then what we found that was interesting is cultivarulose richness was actually important for FMT efficacy. If the donor's cultivarulose richness is higher than that of recipient, which is shown in the figure, that we compare the donor and recipient. If the donor is higher than the recipient in terms of the cultivarulose richness, then all of them were cured. Well, if it's lower, then that is not the case. And for those patients, if you use a donor that is with a high cultivarulose richness, the patient response did well, and over time their cultivarulose richness was sequentially decreased during follow-up. And then we looked at whether donor viral transfer can actually associate with the treatment response. And again, coming back to the donor-recipient effect, if the donor's richness is high than the recipient, then there's a high probability of cultivarulose transfer between donor to recipient. And for those who responded to FMT, they have definitely a higher colonization rate of the cultivarulose that were from the donor. And then we also looked at whether this transplantation or colonization is stable over time. Just like the purple bars showed that actually it is stable over time, especially in the responders. Well, in non-responders, there is a lower colonization rate for patient 9. The patient that I said, it has this patient had a late relapse. There's basically a very little of cultivarulose viruses that were transferred from the donor to recipient. And we also compared the bacterial colonization and the viral colonization. From the left-hand heat map, you can see that the bacteria actually can colonize well, but for patients 7 and 9 who failed FMT, they actually had a very low amount of bacterial colonization. But for patient 9, they had a good bacterial colonization, but like I showed you, the virus did not colonize well. So this patient had a late relapse. So we think bacteriophage might be key in the efficacy of long-term FMT outcome. And for this part, we can see that CDI was actually associated with a gut virome dysbiosis, and viral transfer is actually associated with FMT outcome. And donor virome cultivarulose richness is associated with a favorable FMT efficacy. And then we were recently invited by a Nature Reviews, and Nature Reviews, and Lancet GastroHub to review the role of gut virome in fecal microbiota transplantation. And phages, the first role for phages, they can regulate the gut bacterium composition, and then the bacteriophage can also attach to our intestinal mucosa and therefore defend against the pathogenic bacteria. And then phages, there is also a paper published in Cell Host Microbe showing that phages can directly be transcytosed to the lamina propria of the intestine, then it can elicit proper immune responses than either direct that into a pro-inflammatory response or anti-inflammatory response. And then following that, there are studies showing fecal viral transfer or fecal phages transfer in different diseases such as graft versus host disease, obesity, antibiotic-induced microbiome dysbiosis, and necrotizing enterocolitis, stuff like that. Then I want to talk about a little bit about inflammatory bowel disease. This is also an GI disease rising in East Asia, especially in China. Then in 2015, there is a study showing a fecal virome alteration in FMD, where they showed cardiovirilis was increased in IBD patients' phases. But we want to look at the virome at the mucosal level, the intestinal mucosal level. So we recruited patients with UC and looked at the rectal virome at the time. And you can see from the figure that the cardiovirilis was again increased in GI mucosa, which was more pronounced in patients with inflamed mucosa. So we think the increase of cardiovirilis virome might be a characteristic for broad GI inflammation, because this is also observed in C. difficile infection, then that is acute GI inflammation. Now, IBD is a disease with a chronic inflammation, so we think cardiovirilis might be key. And then going back to the species level, the family level, we found the Escherichia phage, enterobacteriophage, actually specifically enriched in rectal mucosa. And then these phages can release, after lysis, they can release their DNA in the and can be recognized by toll-like receptor 9, and then elicit interferon responses and direct the host response to a pro-inflammatory state. And then how about FMT in this therapeutics in IBD? Actually, FMT is very effective in treating CDI, but in IBD, the response rate is only 30 to 40%. Well, there was a study showing if you combine multi-donor together, then the FMT is highly likely to be successful. This is kind of in line with our previous observations in CDI, because the donor's cardiovirilis richness, if it is high, then it is highly likely that the FMT will succeed. Then in this case, in IBD's case, if you combine multi-donors phases, then the cardiovirilis richness is again increased. So that might partly explain the high efficacy observed in multi-donor combination practice. Then how about eukaryotic viruses in IBD and FMT? They used only 9 patients, but they found that in IBD patients, there actually a high amount of eukaryotic viruses. And we think eukaryotics are actually detrimental. And just because you have a high amount of eukaryotic viruses, the FMT response rate tends to be decreased with the increase of eukaryotic viruses. And then we also looked at graft versus host disease, which we only had one patient who is only 14 years old at the time with myelodysplastic syndrome. Then we used bone marrow transplantation But after bone marrow transplantation, this patient severely developed severely graft versus host disease, mainly manifested in the GI tract, where this patient has significant diarrhea and mucosal inflammation. Then we sequentially give him 4 doses of FMT over 1 year's time frame, 1 month time frame. And over time, you can see the eukaryotic viruses will actually decrease, while the prokaryotic virus, coronavirus, will over time increase, because at baseline, the bacteriophages amount is very low. But for this specific patient, the eukaryotic viruses are very high. So we think gut virome is also important in graft versus host disease. And in future practice of FMT in clinical setting, one way we need to focus on how to optimize FMT. We need to focus on selecting proper donors with a high coronavirus richness and low eukaryotic viruses. And for mechanistic and translational research, we need to figure out what viruses or what phages are actually a bad one, good ones, and how to eliminate the bad ones, and what are the mechanisms for them. But this field is still growing. We are still because this is a burgeoning field, and we are still learning and how to do it. For this work, where I need to acknowledge my lab members, most of them are PhD students, and my postdocs and my collaborators from CUHK and Sun Yat-sen University. Thank you all.
Thank you. We'll have a question from floors first. Oh, yes, go ahead.
Great talk, Tao. So I may miss, what is the biggest difference between prokaryotic and eukaryotic virus you treat? What is the biggest difference, characteristics?
You mean the characteristics or the functions?
Oh, yes.
There's a very small amount of eukaryotic virus, but not like SARS-CoV-2. The eukaryotic viruses are very detrimental because they can directly interact with the mammalian host. But the prokaryotic viruses, most of them are phages. They are most milder because most of them cannot directly interact with the mammalian host, but they can interact with the bacterium. The bacterium are important regulator of the mammalian immunity. Therefore, the phages can interact with the bacteria, then come down to the intestinal mammalian immunity. So therefore, phages are milder, but because of the high number of species existing in our gut, so the potential is much higher than the eukaryotic viruses in terms of regulating the immunity system.
Okay, thank you.
Oh, go ahead.
In case of microbiome, we can analyze 16S RNA.
Yeah.
How do you analyze the virome?
Because viruses, they don't have a consistent region or conserved region that we can use to explore them. But we have to, for this part, for studying virome, you have to use metagenomic sequencing. You enrich either you enrich or do not enrich the viral particles in the gut. Then you do just in a bulk metagenomics to look at the viral genomes. Not like 16S, you can query the marker of bacteria, but for viruses, they don't have a very apparent marker. So we have to use bulk metagenomics.
Well, great talk. One very short question. The caudovirus you described, so do you have an idea what specific bacteriophages are responsible for the host? I mean,
Yeah.
the virus is right now the scale is too big, so I wonder whether
Yeah, sure.
you're phage specifically target, for example, Fusobacteria or some other pathobiome types?
Yeah. Thank you, for Professor Kim. So, the coronavirus is only an order of viruses, and because the international viral community, taxonomy community is still debating which viruses we should place it. Not like the gut bacteria, we can go down to the species, but viruses, they are highly divergent, such as SARS-CoV-1, SARS-CoV-2. But now the field is evolving. We are going back down to the species level, and we have now found some phages whose hosts are Bacteroides and E. coli and Bacteroides Cytomicron. For this specific bacteria, we are isolating the phages, but we are still trying to collect taxonomy in terms of the ranking. But at the time, because the study we conducted was 2 to 3 years ago, so at the time we are still not going to be able to understand which the exact taxonomy it is. But now we can do it. But the paper is not published, but we're still doing it. Thank you.
Thank you very much, Professor Taoju. And the Professor Ji Byung Park from I화여자대학교 University take over the second part of the session. Thank you very much.
네, 안녕하세요. 이화여대 박진병입니다. 지금 세션이 굉장히 달궈지고 있다고 생각이 드는데 앞에 세 분 연사분께서 너무 좋은 강의를 해주셔갖고 저도 너무 집중한 나머지 좀 열이 나기 시작하고 있는데요. 그래서 앞에 세 분 연사분께 다시 한번 감사드리고 그럼 나머지 두 분 연사분 소개해드리겠습니다. Let me introduce Dr. Kwang Hee-hong. So he is the head of CJ바이오사이언스 Discovery Center in Korea. So Dr. Hong received a bachelor's degree in biochemistry from Yonsei University, followed by a master's degree in bioengineering from KAIST. And he obtained his PhD in biochemistry from Brandeis University in the United States. So please give him a big applaud.
Thank you very much for a kind introduction. I'd like to start my presentation by thanking the organizer for inviting me and giving me this opportunity to introduce microbiome drug development strategy at CJbaiosayense. This is the contents. I'm going to give a little bit of background information about microbiome and CJbaiosayense. But regarding microbiome, we've been hearing so much about the introductory stories, so I'll just briefly touch on. After that, I will talk about some strategies in the company, including core capabilities and R&D pipelines. So what is microbiome? So especially for the human microbiome, collectively refers to all microorganisms living in the human body and their genomes. And it is well known that it's very closely related to the human health. Microbiome consists of 4 types of organisms: so bacteria, fungi, viruses, and archaea. So it is known that the number of bacteria living in the human body is slightly more than or similar to the number of human cells, which is around 30 trillion cells. And as shown on the right side of slide, there is a correlation between microbiome and human diseases. So there are many terms to describe the functions or the relationships between microbiome and human organs, like a gut-brain axis, or gut-lung axis, or gut-skin axis. There are a bunch of evidences, scientific evidences, to support that this kind of correlation between microbiome and human health. As shown in this slide on the left side, that the microbiome was discovered to control and influence the efficacy of diabetes medication called metformin. On the bottom right side, this is an interesting story. There's immune check inhibitors are widely used to treat cancer patients. And there are 2 types of cancer patients who are responders or who are non-responders, which means immune check inhibitors doesn't work for such kind of patients. In this experiment, especially for the melanoma patients, the feces from responders were transferred to the non-responders. And then the non-responder patients became susceptible to the ICI. So this says that there's something in the feces. We think this is a microbiome. It's doing something to change the efficacy or the function of the drug. So the many companies are trying to develop that as a new modality, the microbiome as a new drug. So currently, we think around 10 clinical assets are being under development by 50 companies worldwide. As shown in this slide, the focus area is oncology or gastrointestinal or infectious disease. So most of these kind of diseases are related to the immune functions between microorganism and host cells. And on the right side, the microbiome drug development industry is still early stage, so the market size at the moment is small. However, as time goes, more microbiome drugs will be developed and commercialized by approving government authority, we believe. Then it has huge potential to grow in terms of industry market size. On the left side is already, it was as an introductory session in this session, the two products were approved by US FDA, that is Ferring Rebiota and Serigis Vos. Both are for treating the C. difficile infection. This is a very important milestone and event in the microbiome drug development industry. Because of these two drugs approved by US FDA, some doubts about the possibility or the feasibility of microbiome drug development were relieved. However, this is still FMT type of drug, which means in order to produce the final drug product of those two drugs, we need donors' sample. Therefore, this is a little different from traditional pharmaceutical drug product manufacturing process. And also, this is a complex of donors' samples, so we don't know, it's not clearly defined what's in there. So in terms of quality control or other things, there's some limitations. So many companies are trying to develop like a pharma biotics type of drug development as shown in this slide. So only a small portion of FMTs are under development tried by companies, but mostly shown in blue color, like consortia or single strain or engineered bacteria as a main portion for the drug development worldwide, and in different indications altogether. So this is just a short, the picture of the microbiome industry, and from now I will talk about CJBioscience as an introduction. CJBioscience was established about 2 years ago, the beginning of 2022, by combining two companies. One company is 쑨원대학교, so one is a Red Bio department of CJ제일제당. CJ제일제당 is everybody in Korea knows well. It's a food and company. CJ제일제당 acquired the 쑨원대학교 in 2021. 쑨원대학교 was established in 2009 by Professor Chun in Seoul National University. Ever since, 쑨원대학교 collected all the microbiome data to set up a well-curated database. Also, is specialized in bioinformatics. So we think the capabilities and functions 쑨원대학교 is essential for the drug development from CJ제일제당's point of view. So we acquired and then we established CJBioscience by synergistic combination of 쑨원대학교 and the Red Bio sector of CJ. We have a headquarters in Seoul and R&D center in Gyeonggi Province, CJ Blossom Park. Also, we have a subsidiary company called EasyBiome in Maryland. As I mentioned, the 쑨원대학교 has particular, core capabilities in the microbiome, big data, also bioinformatics. And CJ제일제당 has development capabilities. With this synergistic combination, we think our vision is to become global #1 microbiome company. We are very serious about microbiome drug development. Before I go into actually the strategy in CJBioscience, this is microbiome is a new modality, the characteristics of a microbiome-based drug. This is a modality, and then most of the sources to make drug is from healthy donors. We screen and purify and see the non-clinical and clinical efficacies with this strain which is derived from healthy donors. So the safety is not a major concern. So I think this is a very good part. And also we are aiming for treat unmet medical needs. And it also has a great potential in preventive medicine and healthcare, also expanding to other business areas. And also regulatory body, especially US FDA, also Korean MFDS is very keen to set up a new guideline to set up a microbiome drug. So it's LBP, it's a live biotherapeutic products, is as a new category they classified. So with that, I'm going to talk about a little bit more about CJBioscience's strategy. By showing this slide, CJBioscience's core capabilities can be explained only on this slide. We have a platform, it's called the EasyM platform. And in order to discover and develop microbiome drugs, all these essential functions are needed, like a strain bank. We have more than 10,000 strain banks, and around 30% of from UK-based company, 40 Pharma. We acquired 40 Pharma's all the assets made this year. Also, I'm going to talk a little more about regarding this acquisition later on this presentation. And also we have capability for screening for efficacy evaluation. Also, of course, a non-clinical research system, also bioinformatics research system. Also, we have a small pilot scale of a manufacturing facility to do process development and manufacturing, especially for the anaerobic bacteria. Most of the sources for our drug candidates are from gut. Therefore, those are strict anaerobes. So it is some hurdles to manipulate the anaerobic bacteria into the larger scale DS production and finally the DP production. Also, we actively do cohort study and also do some clinical studies. All together, these are all the essential parts for the microbiome drug development, and I must say, CJBioscience has all these functions, is fully integrated discovery and drug development platform that CJ owns. With that, we can do drug development. Also, we can expand to the business areas. So actually, we are also doing the NGS service business in our company as well. And this is our business model. So we do from discovery status. So we do actively discovery status and preclinical studies and phase 1 and phase 2 clinical studies. Once we do the phase 2 clinical studies to prove the proof of concept in human clinical studies, then we'll actively look for the opportunities to collaborate with the multinational companies. In some indications, some other candidates, if we think, enough data in the stage of non-clinical stage, we can also do a partnership with other companies. On the basis of all these activities are our platform technologies are involved. So this is overall picture of our strategy. Our main focus area for drug development is 3 areas. So one is immuno-oncology, especially for lung cancer, and the autoimmune disease like IBD or respiratory disease. So many previous talks talk about IBD and other diseases. I know that many microbiome companies are trying to develop drugs for the IBD category. And for the CNS, especially for Parkinson's disease, so those are the categories that we're focusing on in terms of therapeutic areas. We've been actively trying to expand our R&D pipeline by doing open innovation, which is collaboration with other companies, or the in-licensing activities to acquire the pipelines, so that we were able to acquire UK-based company called 4D Pharma in May this year. The 4D Pharma's strategy is quite aligned with our strategy in terms of modality and also the focused area. So they were focusing on immuno-oncology, CNS and immune-related disease and gastrointestinal diseases. Also, they were trying to develop the single strain-based LBP, which is the same as ours. So because of that, we decided to acquire all their assets, and including all the R&D pipelines and intellectual properties as well, and also their bacterial strains and other documents and all of the assets. So all combined together, our pipeline goes like this. In total, 15 R&D pipelines currently we have, and 4 of them shown in blue color are in-house developed pipelines, and the purple colors are from 4D Pharmas. We reviewed all these documents, their data all together, and prioritized among these pipelines which one will be developed first. Strategically, we decided from among these pipelines. Okay, so that's the current status of pipeline, and from now I will talk about one of our leading program called the CJLB-101. CJLB-101 is a clinical stage program. This is a microbiome-based immunotherapy candidate for NSCLC in combination with pembrolizumab. This is a novel strain and a single live biotherapy product candidate. And we have received IND approval from US FDA in the beginning of this year for phase 1/2 clinical trials for anti-cancer agent, and from Korean MFDS in June of this year. We already started our clinical study in Korea, so in the later part I will talk about our R&D, clinical trial design altogether, some status of current development. So now, next to go into some non-clinical efficacy study data, and this one is important findings for our candidate efficacy. This is an in vivo test, the mouse model and PDX model, so patient-derived xenograft. And on the left side, the y-axis shows the tumor volume, and the x-axis shows days after treatment, so that when the tissue, the cancer tissue from patient was implanted into mice, shown in this black solid line, as time goes, the tumor volume is increased. However, on the contrary, CJLB-101 treated group, shown in red color, showed a strong anti-cancer efficacy, as the tumor growth was inhibited by administration of our drug candidate. We performed the same experiment on the right side. However, as you can see, the red color did not show any efficacy in this slide. So what is the difference between those two experiments? The difference is the mouse strain that we used for this experiment. The one on the left side, it has a humanized mice, and it has immune system. It has a human immune system inside it. But the one on the right side, which did not show any efficacy, is immune deficient mouse strain. Therefore, this clearly tells that our drug candidate exerts its functions via somehow human immune interaction. So the anti-cancer efficacy of CJLB-101 is T-cell dependent immune responses. So in terms of efficacy and also in terms of seeing the mechanism of action, this is an important result. And next one is representative non-clinical data, putting them together, showing the same type of experiment, the PDX mice model, and then we put different groups of the samples, like pembrolizumab shown in blue color, and the red color CJLB101, and the yellow and dark red are a combination of CJLB101 and pembrolizumab with the different dosages. So this waterfall plot shows that the TGI level, if the bar graph goes to downward, the bigger the bar is, the higher the efficacy. So showing that almost of the red color, the CJLB101 single agent or the combination groups are showing the strong efficacy. If you look at the right side of data on the center one, I'd like to emphasize this one especially, because this type of the cancer tissue was derived from refractory patient, which means this patient, although the pembrolizumab was administered, that didn't work. We called it pembrolizumab refractory patient. So pembrolizumab refractory tissue was implanted in the mice. Therefore, shown in this, the black line is negative control, but the gray line is pembrolizumab administered group. Obviously, this is pembrolizumab refractory cancer tissue, showing that there is no efficacy even though pembrolizumab was administered in the mice. But in the case of CJLB101 single agent administration or the combination of the administration showing the strong, robust anti-cancer efficacy. So this is a very important result because Keytruda, the trade name of pembrolizumab, is a globally top-selling product. Very good, excellent, top-selling product. However, it is known that 1 out of 4 patients are only responders. The remaining 75% of patients are non-responders. However, from this experiment, we think this has a huge potential to treat pembrolizumab refractory patients with the combination therapy or the CJLB101 monotherapy. So that's the efficacy data. So here I will show some mechanism of action, the mode of action. So the macrophage, which is important for the innate immune response, is important to show the anti-cancer efficacy of CJLB101. From this type of experiment, we did some differentiation trajectory of macrophages, showing that by administration of CJLB101, the M0 type of macrophage was polarized to M1 type of macrophage. Also, already polarized to M2 type of macrophage is repolarized to M1 type of macrophage. Those two events increases the level of M1 over M2, and also we do single cell RNA sequencing to see the expression of chemokines, showing that the CXCL9 and CXCL10 were highly expressed in that M1 macrophages. With that, we know that M1 type of CXCL9 positive and CXCL10 positive are actively involved in the further immune reaction, which can recruit and activate the CD8 positive T cells, which is cytotoxic. So that we also know the level of interferon gamma was increased, which then exerts the anti-cancer efficacy in our experiment. So put them together, the mode of actions, when you take CJLB101 orally, although I did not show some evidence, but we know that some cell surface component of CJLB101 is important to exert efficacy. And then cell surface component is somehow interact with the host immune system, then induce the polarization and the repolarization of a macrophage, and then subsequently to the activation of T cells in around the tumor microenvironment, showing that efficacy. And this is the final page to show the clinical trial design. So we start from low dose plus, this is all combination therapy with the Keytruda. So 20 patients in low dose and high dose and 20 patients all together is phase 1 clinical trial. And for the phase 2 clinical trial, this is planned. So October 13th, we enrolled the first patient and administered our drug combination with Keytruda and monitored for 3 weeks whether there any other safety issues are there. So no concern, no safety issues are revealed. After that, the second patient was enrolled and administered our drug combination, and then we monitored 1 week. That's the regulatory guidance. First, this is ever since human trial in the first time, so the regulatory body asked us to monitor at least the first patient 3 weeks and second patient 1 week. After that, if it's okay, then we can do simultaneously recruit many patients as well. So it's been going smoothly, and we are hoping to have a good result from these clinical trials. Thank you very much for your attention.
Thank you very much for a great talk. So it's time for discussion. So any questions from floor? Yeah.
That's very interesting talk. Could you tell us about the JB101?
Yeah, CJLB 101.
Yeah, more. You told us it may be the key materials to do function of this bacterium. Maybe a surface something on this bacterium.
Yeah.
So, do you have any clue what is this surface materials?
What is it? We don't know yet. However, we did perform some fractionation experiment.
Then it may be some metabolite even, right?
No, not metabolite. But the cell surface molecule, like a cell membrane component,
I see.
Yeah, some lipid or polysaccharide or some other things. So we don't know exactly
Okay.
what works for that.
So-
However, we did perform fractionation experiment. Cell wall component, the cell membrane component, and cell metabolite, the cell lysate. So we did whole different types of fractionation experiment. We have a very strong evidence that the
Yeah.
cell membrane is the
Yes,
one.
you also you told us this promising bacterium activate to M2 paste to M1 paste,
Yeah,
right?
yeah.
So did you check the any inflammatory cytokine, not interferon gamma?
Yeah, we checked a whole bunch of cytokines, everything.
So
And we saw the IL-6 elevation, IL-10 decrease. And also TLR4 is important receptor for
So,
mediating all
yeah,
these
this
actions.
is for anti-cancer
Yes.
therapeutics. That's why. Okay, thank you.
Yep.
I have also one quick question.
Yeah.
How would you make cells lysed? So did you use enzymes or did you use chemical lysis of cells for preparation of the cell metabolites?
The
Fragments.
exact methodology, maybe I'm not sure. I can say that this is a heat-killed bacteria is working, and also some physical lysate and also supernatant when you grow the bacteria. That's what we try to as a metabolite portion. That's how we separate cell portion and metabolite portion.
Okay, so thank you very much.
Yep.
So, yeah.
Thank you.
So let me introduce the last speaker of this session. So Professor Suryang Kwak, he is a professor of Kookmin University. He received his MS and BS and MS from Seoul National University from Professor Jin Ho Sir lab, and he moved to Dr. Yong-Soo Jin's lab at the University of Illinois. Afterwards, he also studied at the Professor Gautam Dantis at Washington University School of Medicine. Please give him a big hand.
First, I very much appreciate this opportunity to share my research today and your kind introduction, Professor Jin Byung-Park. And thank you all for being here at the last moment of this conference. I will introduce. Actually, I didn't know that the previous lecturers, almost all lecturers discussed about C. difficile infection. So I included this introductory slide, but maybe I will skip this not skip, I will touch very briefly. C. difficile is the main pathogen of the studies I will present today. And its name was Clostridium difficile, but its official name currently Clostridioides difficile. And it's a representative opportunistic pathogen. And C. difficile infection, the CDI, is one of the most common nosocomial infection causing diarrhea. And it's estimated to cause nearly 500,000 infections in the United States each year. And around 20% of the previous CDI patients will get it again in the subsequent 2 to 8 weeks. And it is known that CDI is usually caused by damage to the healthy intestinal microbial community. And actually this picture explains the situation very well, so I included this. So when the structure, when the structure, of the commensal microbiome in our gut is damaged, usually by antibiotics, and then C. difficile actively flourishes in our gut. And the antibiotics are known to be the main cause of microbiome dysbiosis, as I just mentioned, and the C. difficile can withstand multiple antibiotics in its spore form. That's the problem. And let me explain a little more about the relationship between the antibiotics and CDI. The Shannon index, in this figure, the left figure is one of the alpha diversity indices, and the index value of the gut microbiome of healthy individuals is generally higher than the dysbiotic microbiome, usually found in the patients prescribed antibiotics in hospitals. Actually, this experiment was conducted on murine model, but humans also show similar patterns. And many of the mice, because this is a murine model experiment, many of the mice with the dysbiotic, the yellow microbiome, maintain the number of C. difficile cells in the stool for more than 6 days. But regular mice with just okay gut microbiome reduce the number of C. difficile cells relatively quickly. This is the colonization resistance. The higher alpha diversity, the stronger colonization resistance against the C. difficile. So antibiotic treatments damage colonization resistance, as we just discussed, by the gut commensal microbiota. That is the most important risk factor of CDI in the hospital. And it is ironic that antibiotics such as vancomycin and metronidazole or fixadomycin, fidaxomicin are still the first-line therapy for patients with CDI. So therefore, many previous CDI patients experience recurrent CDI, RCDI. So we need alternatives for the current antibiotic-based treatment to control CDI. So in today's presentation, I will introduce 2 representative approaches, alternative approaches that I have tried. First one is, you already heard so many times here, fecal microbial transplantation. And second is prebiotic, especially engineered prebiotic biomaterials. Originally, I should briefly introduce about FMT, but already many lecturers, multiple lecturers, actually all the previous lecturers discussed about FMT. So but FMT introduces fecal microbiota of healthy people into patients with gut microbiome dysbiosis. And this can be a great alternative to antibiotics for infectious disease, especially because the healthy microbiota usually exhibit higher taxonomic diversity, and it would be great if the FMT can recover the dysbiotic gut microbiome that originally allows blooming of opportunistic pathogens like C. difficile. And healthy people are not exposed to antibiotics like patients in hospitals, so usually exhibit lower ARO and ARG, antibiotic resistance organism and genes, relative abundances levels. So many previous studies also argue that FMT even can decrease the antibiotic resistance level in the gut. Recently, FMT for the C. difficile infection was approved by FDA. Just the CJbaiosa이언스 already introduced this fact, but yeah, around a year and a half ago, the efficacy and safety of FMT was still very controversial because first there was no previous FMT study performed with placebo controls, more specifically the metagenomic studies. Before the study I will introduce soon. And another concern is about antibiotic resistance organism and genes, ARO and ARG. Although most FMT donors are very thoroughly screened healthy people, but still a death case of a CDI patient could happen due to bacteremia of MDRO, multi-drug resistance organism, E. coli introduced by FMT. So actually the FMT studies were shortly stopped by the recommendation by the FDA previously. And lastly, we still do not know clearly how FMT cannot be successful for certain patients or successful for other patients. Not every FMT recipient shows successful clinical outcomes, although it is at least better than placebo, as you can see here. To properly evaluate how FMT really affects the microbiome diversity and abundances of AROs and ARGs, we analyzed and tracked stool samples from the patients with recurrent CDI who had joined the clinical intervention of RBX2660. Actually, this is the Rebiora of the Ferring company. And actually, Ferring's the Rebiora is the fecal microbiota biotherapeutic, as it just was introduced, that FDA recently approved. And these samples were collected during phase 2 clinical trials of the Rebiota, exactly, specifically RBX2660. And very fortunately, this study was conducted in randomized, double-blind, placebo-controlled design at multiple international clinical centers. And this study has 3 cohort groups, most importantly including the placebo recipients and also double-dose recipients. So we could track the clinical efficacy of FMT or safety of FMT in dose-dependent manner. And patients' stool were collected for up to 1 year from the day of the first FMT. The first take-home message of this study is that FMT actually was not the reason of the recovery of diversity. The healthy microbiota, in this case, the RBX2660, indeed showed significantly higher taxonomic diversity compared to the patient's baseline before FMT treatment. But all patients, including the placebo recipients, showed the increasing trend of taxonomic diversity. Still, FMT transformed the patient's gut microbiome taxonomic structure toward those of the donors in dose-dependent manner. So actually, FMT works regarding the transformation of the taxonomic structures, but regarding alpha diversity, actually it doesn't work at all. So to more precisely quantify the taxonomic transformation at this section, we developed a simple index at the bottom. It's namely transplantation index based on the Bray-Curtis distance, dissimilarity between the patients and corresponding donors' microbiome, and showed that taxonomic transplantation by FMT was indeed dose-dependent, especially for the first week after FMT, the first data. And we found that a species named Bellonella typica, the middle data, which is known as a great gum microbe of Elithosaletics. This strain is very well known as which have the capability to change the lactate fatigue material to the energy material propionate. And it was the biomarker of a successful taxonomic transplantation within the first week. And some patients, however, even after getting double dose and with waiting for 60 days, their microbiome taxonomic structures could not be transformed by FMT at all. And their microbiome has higher abundances of innately vancomycin resistant organisms, the right data. So, and vancomycin was the major antibiotics for CDI. Actually, currently still, it's the major antibiotics treatment for CDI patients, and in this study as well. So this means that the patients who showed delayed taxonomic transplantation by FMT previously got more dysbiotic microbiome shaped by the vancomycin. And then we delved into eradication of ARGs or AROs by FMT. Actually, there was no FMT effect on the ARG abundances as well. We assessed the registome, namely the all ARGs in each patient's microbiome, and healthy microbiota RBX 2660 in our case exhibited significantly lower the relative abundance of ARGs compared to the patient microbiome, especially at the baseline. However, the FMT did not decrease ARG at all, as you can see here, like the taxonomic data. Placebo also decreased the level of ARG abundances over time after FMT, like IBX 2660 recipients. And these bar graphs represent the average registome structures of the patient in each group at each time point. And as you can see here, placebo recipients showed similar registome structural transformation to those of the FMT recipients, regardless of those actually. And however, the registomic transplantation, the right data, was positively correlated with the taxonomic transplantation, and the FMT resulted in more active taxonomic and registomic fluctuation than placebo. And these facts imply that FMT can introduce new ARGs together with the new microbiota. So it's still FMT is effective, but maybe this can be a potential risk of FMT. And this is another little concerning fact. We also tracked the patient-derived baseline-derived AROs and donor-derived AROs, organism level, not gene level, and discovered that FMT really more effectively eradicated patients' baseline-derived AROs. It's effective, but at the same time introduced the new AROs. These red, I'm sorry about I cannot show the cursor, but yeah. These red dots are the newly introduced AROs, and most of them are, actually all of them are ARO E. coli, and actually one of them was MDR E. coli. And actually the introduction of new AROs did not affect the clinical efficacy of FMT. I mean, the repression of another episode of CDI. I mean, this newly introduced ARO didn't affect the clinical efficacy, but still, this can be a little concerning point. Yeah, the summary about the first section, although FMT has been known as an avenue, the recovering damaged diversity of recipients' gut microbiome and eradicating antibiotic resistance from the gut microbiome. But our placebo-controlled study revealed that it is not 100% true. FMT is still effective against the C. difficile infection, but previous assumptions are not true. Some of them are not true, and probably discontinuation of antibiotics during the FMT clinical trial, because during FMT clinical trial, the doctors stopped the administration of antibiotics because it's not compatible each other, and the discontinuation of antibiotics might be the major cause of the misunderstood benefits of FMT so far. And we found this. I actually intentionally omitted several data because it's not published yet, and I was still am working with the parent company and Washington University's School of Medicine. It's not appropriate to open the data yet. But one thing I can say is that actually Akkermansia mucinipilia, we already discussed multiple times here. It's not very well known positive strain in our gut, but actually the presence of Akkermansia mucinipilia was a negative factor of successful FMT regarding the repression of ICDI. But still, anyway, the backend mechanism determining the success or failure of FMT against the CDI is in a black box. So actually, the conclusion of this first study is that both antibiotics and FMT have their own limits, and we may need a better alternative. And that's the reason why I'm introducing this second topic about S. bladii, Saccharomyces bladii, a probiotic yeast strain. S. bladii is already very well-known effective probiotic against multiple intestinal disorders, including C. difficile infection. But instead of probiotic functionalities, I have focused more on its prebiotic functions derived from its cell wall oligosaccharide. Saccharomyces bladii is actually a variant of Saccharomyces cerevisiae, the brewer's yeast, and they share almost identical genetic properties. But S. bladii has higher cell wall mannan content as compared to the S. cerevisiae, its parent strain. And the mannan oligosaccharide has an interesting prebiotic effect. As you can see in this left data, it decreases if we administer the mannan oligosaccharide to mice, then their gut microbiome showed decreased Firmicutes level and increased the Bacteroidetes level. In phylum level, and Bacteroidetes genus is the major momentum of the increase in the Bacteroidetes phylum, and Firmicutes, including the C. difficile, they must chop the mannan oligosaccharide into monomer or dimer levels to assimilate it, but the Bacteroidetes, including Bacteroidetes, for example, Cetiotan or Ovartus, they can assimilate mannan oligosaccharide after cutting it into large chunks that Firmicutes cannot utilize. It's a very selfish way, and I hypothesize that this selfish mechanism allows Bacteroidetes to compete better with the Firmicutes. And maybe this is the major momentum of S. bladii clinical efficacy against the C. difficile infection. So I wanted to engineer this probiotic yeast S. bladii to increase their cell wall mannan oligosaccharide level further to prepare a super prebiotic biomaterial as a treatment against the C. difficile infection. Saccharomyces yeast synthesize cell wall oligosaccharide like mannan oligosaccharide or glucan from the intermediate of the glycolysis. They have a very good metabolic pathway to synthesize it, but Saccharomyces yeasts have a very peculiar metabolic stance, namely overflow metabolism or Crabtree effect, showing very strong and rigid metabolic fluxes toward the glycolysis through glycolysis and toward the ethanol fermentation on fermentable sugar like glucose. So it is very difficult for S. bladii to produce other materials than ethanol, despite its potential to do it. So I omitted another slide here because of the time limit, but I found a very interesting mechanism of glycolysis shunting from cancer cell. Cancer cells can shunt the glycolysis flux by inactivating the phosphoproctokinase, which can convert the fructose 6-phosphate to fructose 1,6-bisphosphate. This is the major bottleneck step of the upper glycolysis. And then the upper glycolysis intermediates can be accumulated. I mean, it's a little awkward. Maybe better expression would be their urban availability will be increased. But in the case of S. bladii, it's polyploidy, so it's difficult to make some fine genomic engineering. So I just deleted this fructose 2,6-bisphosphate synthetic pathway. fructose 2,6-bisphosphate is the allosteric upregulator of phosphofructokinase. And then after the deletion, I found that indeed glucose 6-phosphate and fructose 6-phosphate, the intermediate of upper glycolysis accumulated significantly. And then additional overexpression of GDP mannose synthetic pathway increased the GDP mannose availability significantly. And this can be used for the mannan oligosaccharide on the cell wall, a synthesis of mannan oligosaccharide. And it indeed affected the several mannan oligosaccharide level as well. And by overexpression and deletion of enzymes for mannan elaboration in ER and Golgi complex, I could further increase the several mannan oligosaccharide levels. And interestingly, additional overexpression of mannan elaboration pathways in Golgi actually did not positively affect the several mannan levels. But the deletion of GDA1, this is a kind of like transporter of GDP mannose, and the deletion of GDA1 was not lethal to the Saccharomyces bladii or other Saccharomyces yeast strains, but still very effectively decrease the white bar here in the middle data. Very effectively decrease the mannan oligosaccharide level, so this can be used negative control strain. And same genetic engineering actually was not effective at all in Saccharomyces cerevisiae, but only effective in Saccharomyces bladii. It means that bladii has potential to over-synthesize several mannan or glucan oligosaccharide, but cerevisiae somehow does not. So eventually, I could prepare a set of yeast strains with varied several mannan levels and actually glucan levels as well. And these are the cross-sectional TM images, and this hairy, the outside the hairy layer is the mannan oligosaccharide layer. And the thickness of several oligosaccharides, like mannan layer and the glucan layer, the middle layer, affirms the previous chemical quantification. And wild-type bladii showed already significantly thicker and coarser mannan oligosaccharide layer than S. cerevisiae. And the GDA deletion damaged the several mannan layer regarding both the thickness and the density. And we did the yeast strains. We performed the co-fermentation of bis tau micron and the representative that's a representative bacteria, this commensal bacteria, which can utilize mannan oligosaccharides efficiently, and C. difficile on biomass of the selected engineered yeast strains with varied cell wall levels, several oligosaccharide levels. And we confirmed that we can increase the selectivity for the proliferating bis-tau micron over C. difficile growth by enhancing several mannan. And glucan did not affect the selectivity at all. So the summary of the second study is we engineered the probiotic yeast S. bladii as a prebiotic biomaterial against the C. difficile infection. And I also want to emphasize that all the genetic engineering performed here was performed by CRISPR-Cas9 system without any heterologous genetic elements. So the final construct is not a transgenic strain. It's a non-transgenic strain without any other genetic elements. I just shuffled the homogeneous gene elements only. And in addition, although this is not directly CDI related protective feature, but we also found that the binding affinity of S. bladii cells to pathogens carrying mannan-specific adhesion, like Salmonella enterica type murium here. And it is positively correlated with S. bladii cell wall mannan contents, but not glucan contents. And I would like to thank my previous colleagues and funding sources kindly supported the studies I presented today. Thank you so much.
Thank you very much for your very interesting talks.
Thanks.
So it's time for discussions, questions from the floor.
Very interesting talk. So the mannan that is produced by Saccharomyces can protect against C. difficile. In our study, we found Candida albicans, which is another fungus. On its cell wall, it also has a lot of glycans. So is there a difference between Saccharomyces and Candida? Because they definitely have different roles. Saccharomyces can guard against C. diff, while Candida albicans maybe exacerbate C. difficile infection.
I thank you for the great question. At least I can say that the Candida strains also have a monooligosaccharide, but maybe the exact structure of oligosaccharide, like monooligosaccharide or glucan, on the cell wall should be different. And frankly speaking, the only reason why I picked Saccharomyces yeast strain to engineer as the prebiotic biomaterial is that Saccharomyces is a well-known and aggressive strain. It's a safe, well-known safe strain, especially as Bladia is a well-known probiotic strain. That's the reason. Maybe I misunderstood your question, but I think if we will kill the Candida and Aspergillosis cells, and I mean just quenching all the biological activity of the both yeast strains, and then maybe C. difficile or B. cetal tau micron utilize them as the carbon sources, just almost identically in my opinion, but because they actually have a different bioactivity in our gut, in addition to as a carbon source, I actually no idea how they will change the differences in the gut, host gut.
Okay. So the other question is, many is actually a ligand to dactin, like toll-like receptor 4. It can evoke a pro-inflammatory response. Did you check whether many can, though the phenotype tells us that it is protective against inflammation, but did you do any in vitro, in vivo study to look at whether the mannan produced by the yeast can actually evoke the dactin signaling pathway?
Great question, thank you. Unfortunately, I didn't try any animal cell-based experiments or mouse model experiments for the engineered yeast strains yet. I'm still further engineering the presented Aspergillosis cells, and then maybe after completion of the construction, then maybe I will check the immunological effect of the engineered strain and wild type strain, as you just mentioned. But yeah, sorry, I have no idea yet.
Thank you. My last question, just being curious. So, is the Saccharomyces strain that you were using for engineering is approved by your government? Because it is not approved by China government, because we're using bacteria, and even though the bacteria that will approved is only a handful, but we don't approve any yeast-based probiotic. For the strain that you are using, is it approved by Korean government?
I don't think so. But in the case of United States, it was approved, as far as I know. Yeah, Bladia and Therapeutics both are approved to be engineered like biotherapeutic host system. Yeah, that's what I know.
Okay, thank you.
Thank you.
Yeah. Let me finish discussion here, so because of time limit. So please give Professor Kwak him a big hand.
Thank you.
네, 이상으로 이번 세션에서 열강을 해주신 5분 교수님, 박사님들께 다시 한번 감사드리면서 이번 세션을 마무리하도록 하겠습니다. 감사합니다.